Therapies for retinal blinding diseases. We do that by targeting directly the photoreceptors, and we do that in a gene-agnostic fashion. So we initially target retinitis pigmentosa, which is the leading cause of genetic blindness, but we also can expand, because of the biologies we're using, expand the application of these products to other retinal diseases, and notably macular degeneration, age-related macular degeneration. The biology we're using is twofold. One, we provide neuroprotection directly to cone, and also at the same time thwarting any oxidative stress that happens in those blinding diseases. The second product we're using is neurorestorative. We basically give back the potential and generate a signal that can be transmitted to the brain for vision interpretation. A signal that can be transmitted to the brain for vision interpretation. We just completed our first clinical trial on Prodigy with the first product, the neuroprotective product. Completed as in finished dosing the patients. The second for SPVN20, the neurorestorative product, is currently being enrolled. We also have the potential for a combination of the two products in a single product, ultimately to provide both at the same time neuroprotection and neurorestoration. So I'm going to really go back to AAV biology real quick, because that's important for what I'm going to be talking about. AAV is a small dependovirus. It's non-pathogenic. It's used widely in the gene therapy field because of all its properties. It's easily manipulatable. It persists in non-dividing cells very long. It provides very good expression. It's a very simple virus initially, a capsid encoding a piece of DNA. And you have here the two natural rep and cap gene. The proteins of the capsids, it's not this one. The proteins of the capsid come different in terms of naturally occurring, so that provides different serotypes of AAVs, and also those proteins can be engineered in the laboratory to enhance certain type of tropism for certain tissues. That's also important for what I'm going to be talking about. In order to make vectors out of an AAV genome, the rep and cap genes are replaced by a transgene cassette, and we keep the ITRs, the inverted terminal repeats, which is really basically the signature of an AAV genome, and they have their own functions. Moving along, the manufacturing of AAV vectors is through a split genome approach, where the components of the vector system are provided in two or three different plasmids, one encoding the gene of interest cassette between the ITRs, the rep and cap that is only required for the cells producer cell to actually make the capsid, and the helper, as I said, I didn't say it, but a dependovirus requires a helper virus to replicate. So those three components are provided usually in 293 cells in bioreactors for the production of the vector. Purification is classic, like any kind of protein, through chromatography and filtration. I'm not going to elaborate on that. So what's happening in a producer cell, basically, is that once your three components are present, they are expressed or replicated in the case of the genome to fabricate basically the vector that is intended to be produced. However, by way of recombinations, which mechanism is not known exactly, we also find those byproducts here where the different components can recombine and generate those weird little fragments here of genomes, some of which contain back ITRs and rep caps. So basically creating those replication potentially competent viruses. The agencies, regulatory authorities, ask us to test for these, the presence of these recombinant competent products as part of impurities that are occurring as part of the manufacturing. There's potentially the risk associated to those are potentially immunological reaction because of an overload of protein for vectors that actually are not going to produce generate the payload. The classic test, so the industry standard test that is used is this one. So it's basically a cell-based assay where the product is put onto permissive cells in the presence of adenovirus as a helper virus, and then passage over 3 times for 3 rounds of amplification. After each round, a PCR is done on the viral genes to monitor their amplification over the course of the test, of the assay. If there were contamination initially and nothing else, the signal would disappear over 3 passages. If there's an rcAAV, then this signal will amplify. And the product is declared positive or negative per a certain dose that was initially put. So it's not a real quantification, it's just positive or less than per a certain dose of vector put on the assay. It's a very long assay, it takes 5 to 6 weeks to perform, and it has its own caveats, as I'm going to detail in a minute. Because it's a cell-based assay, because it requires infection, we are faced with the difficulty of certain serotype not really infecting very well in vitro. And since HeLAs and 293 cells are the ones that are used largely for this assay, you can see that certain serotypes here, and those are only natural serotypes, and you can think of all the engineered serotypes that have been coming up lately in the field. Some of them are very infectious, some of them are non-infectious. Particularly AAV8, AAV9, who are largely used in the clinic today, they don't really infect in vitro. So what does this mean? It means that for a certain quantification, I'm going to go through the details of how we calculate the tissue culture infectious dose, but basically for an AAV2, for example, you have roughly 800 genomes to constitute 10 infectious units of vector. For AAV8, you need 2 million. So there's like 2,000-fold difference between those 2 serotypes. So what does it mean for the assay itself? Is that you can easily declare a negative when in fact the problem is that it didn't infect to begin with. So we scratched our head and said, okay, what can we do to circumvent this? So as I said earlier, a wild-type AAV genome, the architecture is such that you have a rep and cap gene flanked by the 2 ITRs. And we thought that if we can detect by digital PCR simultaneously in one particle the 3 region joining ITR and rep cap, and the cap ITR in a single particle, then you can easily say there. We have a potential rcAAV genome there. So that's what we did. We performed a digital PCR, so that this is the schematic of the assay. We dilute the sample in order to have statistically in each partition of only a few particles. So when we see a signal, we know it's only one particle. And then we look at the presence of signals for each of the fragments that I talked about, double or triple. And each time we have a triple, we know it's a particle that contains a triple positive, and therefore rcAAV genome. We looked at, the first thing we wanted to do is like, okay, are we going to miss something here? So we decided to look at the size of those regions here, because it is a recombination. It's not exactly what you find in the wild-type virus. We wanted to know, how wide can we amplify those regions that are generated during recombination. And we showed that we can go up to 900, closely to 1,000 base pairs, which largely exceeds the capacity of AAV of 4.7 kb to have the ITR, those region plus a rep cap gene, right? So that we're kind of pretty sure that we're not missing any kind of recombinant there. We also looked at the fact that there are some assays out there that are very similar to what we're doing, but they are amplifying only one region. And what you see when you only looking at RTI rep or rep only or RTI cap, you have an overestimation of the outcome. When you do the 3 at the same time, then you go back to what we think are reasonable numbers. So here's the data that we found across different serotypes. AAV2, AAV8, that came again negative in the cell-based assay, AAV9, AAV5. And basically what you find is that for 10 to the 9 VGs, you have sensibly the same number, which makes sense, right? Because for generation of rcAAV, it should not depend on the capsid itself. So with this method, we've managed to show that, in fact, by triple transfection production, all the vectors are created equal in terms of the capacity to generate rcAAVs. Again, to insist on this fact, we found those triple positive genomes in those two products. Again, by the cell-based assay, the AV8 always comes negative. We looked at the different transfection methods. We looked at different scales. We looked at different transgene and again, capsids, and we find out that there's not a lot of impact of the scale at which again, the generation of rcAAVs is unknown. I mean, the mechanism that generates those rcAAVs is unknown, so we don't really know. But maybe there's something linked to transfection. This batch here was done with a particular type of transfection reagent. Turned out to be a little bit higher, but otherwise it's pretty stable across the different samples that we tested. Okay, so I'm missing a slide. Okay, never mind. Just what I wanted to say is that we looked at the vectors produced by baculovirus. If you're familiar, they require a strange arrangement of rep and cap and also promoters that are specific to insect cells. And interestingly enough, we don't find triple positive genomes in those AAV generated by baculo. So in conclusion, so this triple PCR assay allows for an unbiased detection of rcAAVs in a wide array of vectors preparations. We've shown that it's serotype transgene independent. It provides a quantitative information, so we can say we have so many triple positive genomes per 10 to the 9. And it's a much quicker turnaround assay in terms of practicality than a cell-based assay, as you can imagine, because it's only dPCR. What we want to do now is to think about automation, because it takes roughly 10 plates of digital PCR to test 10 to the 9 particles for 10 to the 12 VG per male vector. So I think people who are developing this is not our wheelhouse. Actually, we're looking for partners to test more products to make the assay more robust, but also to be able to maybe create some automation behind this so that a large number of VGs can be tested. The last thing we need to do is really talk to regulators, because the aim is to replace the cell-based assay currently and come up with specifications that would make sense. As I said, the signal is positive in pretty much every batch that we tested. And I had an acknowledgement slide on the other presentation. I don't have it, but I want to thank my team, Fabien, Baptiste, and Benoit, who helped me tremendously with this.